in the US. So thank you so much, and we'll hopefully you'll keep joining us through the length of the webinar.
Okay, thank
you. Thank you very much.
Bye.
Excellent. I'd now like to introduce our next speaker, Dr. Ted Turlings, who is a professor at the University of Neuchatel in Switzerland, and currently he's head of the Laboratory of Fundamental and Applied Research in Chemical Ecology, which focuses on the use of plant-produced signals to improve crop protection. In addition, he's also the director of the newly established Centre of Competence in Chemical Ecology at the University of Neuchatel. And Ted, can you hear me and join us, and welcome?
Yes, I hear you well. Thank you for that very kind introduction, and greetings from Switzerland. I will be talking about how our efforts to use nematodes against Fall Armyworm, and this is a collaboration between the University of Neuchatel, CABI, and the Rwandan Agricultural Board. So the field trial that I'll talk to you about was done in Rwanda. Next slide. And we'll see here the two players in this. They're both the brains and the muscle behind the research that was conducted. Stefan Toepfer from CABI and Patrick Fallet from the University of Neuchatel, a PhD student who is also in the audience and who can answer some of your questions probably better than I can. Next slide. No need to explain to this audience that Fall Armyworm has become an extremely important problem in Africa. And that's why we do our field trials for the moment, but I hope to extend that also to Asia. Next slide. In Africa, estimates from mainly CABI showing tremendous impacts on the production of maize and probably losses of up to $5 billion US dollars per year. Nematodes have a potential as biocontrol agents against many insects. They're usually considered against root pests below ground. But the observation that we made, and you can see that in the next slide, when we started looking at using this nematodes, next, Alison, our first efforts were actually to try to use nematodes against the Western corn rootworm, next, where we have not just the beetle being a problem, but mainly the larvae that feed on the roots. And we're considering to use nematodes in beads, as you will see in the next slide now. We were able to put nematodes in small alginate beads of about ½ centimeter in diameter, up to 5,000 nematodes that we put to sleep by adding a certain chemical to it. And when we add water, they wake up and they are able to infect those larvae in the soil. So we were considering to use this as something that it can be planted in the soil together with the seeds and then against the larvae. But we also were considering to add feeding stimulants and attractants to it. And while we were playing with that in the lab, as you will see in the next little movie, we were trying this without realizing how important Fall Armyworm would be coming. If you can see, if you can get the video started, yeah. So here you see the small little beads. We add some coloring to it, so they're blue. Here the nematodes are moving, but we can put them to sleep. And we were just playing with it, adding leaf extract to the beads, and we got Fall Armyworm larvae that we are also working on at the time, feeding on it. And if there are nematodes in those beads, within a day or two, the larvae will look like this, completely dead. And they become then the source of a new generation of nematodes that eventually will burst out and can also kill the next generation of the pest. So that's what this video is showing. So this was already done before we started working on nematodes against Fall Armyworm. So that had great promise, and we got pretty excited about possible prospects of applying this in the field in Africa. Here you see Patrick with many curious people looking at what crazy things this Swiss guy is doing there. We were then decided to not use nematodes that we have in Switzerland, but actually go to Africa to see if there are any nematodes in the soil there and that we can then apply effectively against Fall Armyworm, because we don't want to introduce new species or even new genotypes of the same species that may be occurring there. We want to use local nematodes, and if we get the opportunity to do that in Asia as well, we'll do the same thing, look for local nematodes against fall armyworm. Next, so initially, next slide, initially Patrick then designed a number of laboratory experiments. Within these cages he had maize plants with fall armyworm on them, and then applied different treatments, including the beet treatments, but eventually we switched to gel treatments. That's easier to apply. So the same material, alginate, but then as a gel applied to the center of the plants. Next slide. And then look at larvae that were on these plants and see how they survived. So here are the different types of treatments that he applied. Just water, nothing in it, oil and gel without nematodes, and then to the right of each of these two bar graphs you see these formulations with nematodes, and you see that there's, in terms of caterpillar survival, the gel application with the nematodes was the most effective, killing 100% of the larvae that he applied in these cage situations. So this is not the field yet, huh? And then in terms of damage to the plants, you see that that's also very significantly reduced by applying these nematodes. I should mention that the oil application is a commercially available formulation that we used as a comparison to our gel. And you see that we're at least as good, the gel. Next slide. So then he went to Africa with Stefan and collaborated with the people from RAB in Rwanda. They selected 4 different fields to do this experiments. I'm going to show you the average for over all the fields. Patrick emphasized to me still yesterday to mention that these fields were very heavily infested by fall armyworm throughout the season, and therefore were ideal to test this. Here's Patrick putting the gel together in collaboration with the people there, including the nematodes. Next slide. This gel was then placed in these glue guns that can easily, relatively easily, squirt this gel into the center of the plants, as you will see in the next slide. In these, so here you see how the application is done, and at the same time, other plants were treated with different treatments like the ones I showed you before: water with nematodes, oil with nematodes, as well as a pesticide that he used as a extreme control measure. Next. So here we'll see a few slides with the people applying here the gel, selected plants that were treated with the gel and nematodes, and others were treated differently. Next slide. Again, you see that that is a real team effort. Next, Alison. And here you see one of the successes. So Patrick smiling there because there were dead larvae, as you see there, and they definitely looked like they were infected by nematodes. Next slide. And that was confirmed. So here's the same picture with these close-up of the larvae that were dead. And when they took those to the lab, eventually, it's not a very good picture, the nematodes burst out. So they were definitely killed by the nematodes under the field conditions. Next. They then, in all of the different treatments that were used, they were used after 5 days. A certain number of plants were inspected for larval presence, and 10 days another group of plants, looking for the numbers of larvae that were present. They also looked at damage, but I won't show you those results today. And here is then the most critical slide of them all, with the raw results for after 5 days, using the control that was not treated with anything as the baseline, so the numbers of caterpillars found there, and then the reduction in the percentage of caterpillars on the other treatments. And you see the pesticide and the gel were about the same. Actually, the gel is slightly better. After 5 days, a reduction in 40% of the larvae. Let me remind you that it was very heavily infested here, because 40% may not be so impressive. But the pesticide didn't do any better, and they used a very high dose of pesticide. So better than the pesticide almost, and that was also at least as good as the pesticide statistically after 10 days. Oil, a commercial oil with nematodes, and water with nematodes were not as effective. So that brings me almost to the end already of this presentation, to show you that this can be used in the field. This is what would happen if you don't treat the plants with anything. The larvae will eventually completely destroy the center of the plant. And in the next slide, you'll see that this is what you end up actually when you have gel treatments. The caterpillars will get killed long before they do that kind of damage. You see the old leaves have larval damage, but the center of the plant has survived this very nicely, and the plant can grow as it wishes and develop completely. A few simple conclusions. The nematodes have great potential to serve as biological control agents in fall armyworm. We recommend that we do weekly applications. This was only once, but the larvae certainly under those very heavy infestation conditions, circumstances will come back, also move from other plants in there. That was probably the explanation why we didn't see better results. The formulation can be further improved by adding attractants and feeding stimulants. And we will want to focus next also on making this as simple as possible, because we want to use this technology for subsistence farmers, and that also applies for possible application in Africa. My last slide is to then thank again the people that did the actual work. So Stefan and Patrick, together with the staff of RAB, who did all this, and they will continue to do this to make it work even better than it already does. I thank them. One more click. And thank you for your attention. I will be happy to answer questions.
Great. Thank you very much, Ted. Can you hear me?
Yes, I do.
Oh, great. Excellent. So sorry. Look, that was a fantastic presentation. Great photos. And I really liked the video. It worked a bit faster for me than I think it did for you. And perfect. And I quite like this idea of putting the nematodes to sleep. It was nice that you could wake them up again, I thought. But look, do you think there are local nematodes in Southeast Asia that a similar project could be developed?
Yeah, there must be plenty of species present there, and the species that we are working with are considered to be globally present. There's still some discussion about the taxonomy, so whatever defines as a species is a little bit up in the air still. So we would like to get, even if is the same species as we may have in Europe, I would like to get the one from Southeast Asia if we want to apply it there. In order not to introduce any different genotypes that are not natively present. But definitely we will find nematodes that could be effective against fall armyworm also in Asia.
Great, thank you. Here's a question. Has weather, or does weather have any effect on EPN efficacy on fall armyworm? So does the weather play a role?
Yes, they are definitely sensitive to UV light, and drought will be a problem. Under the conditions that they were applied in Rwanda, it was during the rainy season, so there was quite a bit of water, and that is, in a way, is good. That also will, in the case we put them to sleep, that will wake them up because the compound will be diluted. That puts them to sleep. But yes, in this gel, we also plan to put some sunscreen, if you wish, to protect them better against the UV light, but already placing them in the whirl of the maize plants will only protect them against UV. And as you see from the results, they did manage to survive and kill these larvae. They survive for several days, but the weather conditions will have an effect, yes.
Okay, just looking at, I know that you took some time in thinking about how to apply these nematodes with the gun, which looked very easy and quite simple but innovative. For large scale application of gel-based application, is that viable, do you think?
It depends on what kind of farmer you're talking about, but the farmers that we're working with right now, I think for them it would be very easy to do it the way we do it. So they have relatively small farms. They already are willing to go out in the field and kill the larvae by hand. So it's a lot less work to apply the gel. We're still working on different types of formulations that are easier to apply, and also with larger squirt guns that we can apply. But if it's a weekly application as we have foreseen now, just manually by the local farmers, I think this is definitely something that would be.
And so economically, have you done some economic analysis comparing it to other methods?
No, we haven't done exact analysis, but I would say that rearing the nematodes would be at least as cheap as, say, the mass rearing of trichogrammas, and would be less effort. I could imagine that we would have small startup family companies that we could teach to do this. We're far from that, so I'm giving a very optimistic scenario. But what I think nematodes are, if we develop the right technology locally, are relatively cheap to produce. The alginate is nothing expensive, and we could probably make that even cheaper. So I'm quite optimistic that in terms of biocontrol, this would be one of the cheapest solutions to use.
Excellent. And just one question: how do those guns? How good? I mean, they're obviously quite simple to use, and anyone can use them in the field. Did you get some feedback from farmers on how they perceived the use of nematodes and how they perceived the method that you've developed?
I think we'll probably let Patrick answer that in the chat, because I'm not, I think they must have talked to some farmers, but it was mostly the local researchers that they work with. So I cannot give you the real answer to that. What I understand of the difficulty is actually to get the gel into the squirt gun. It's a bit of a messy work. So we're now looking at a different kind of formulation that is easier to introduce into
okay.
that.
And I've got one more question that's sort of related actually to what I've just asked, but it's in the same question. In which instar level is the gel and nematodes the most effective? Is that something you can answer?
Yeah, there were laboratory studies, and I've also done here by Patrick and a master student. As far as I remember, all the larval instars get killed.
Okay. And one more question, because we have to move on to our next speaker, but we've got lots there, so you and Patrick are going to be busy, Ted. Do these nematodes have a positive symbiosis in case of roots or negative symbiosis?
A symbiosis with the roots, is that the question?
I think so. Okay.
So these nematodes do not attack the plants. So these are definitely different from photophagus nematodes. And what we actually have, and this is coincidental, but what we have discovered is actually when in their presence, the roots will respond molecularly, and actually the plants will defend themselves more strongly and induces some more resistance in the plant. Completely different topic, but it's definitely an interesting additional benefit that we think we will have by.
Well, that certainly is an interesting additional benefit, and that's probably a good place to leave it for now. There's lots of questions coming in for you and Patrick, so feel free to get on the Q&A and answer some of those. And thank you very much, Ted, for joining us, and thank you for Patrick too, and some great photos of seeing you out there in the field too. That's very nice to see.